essentially what I'm going to do is go through some of the things that I find more useful in ChimeraX for looking at protein ligand complexes, and it's not an exhaustive overview of everything in ChimeraX, and we will also touch on some of the things to do with looking at AlphaFold structures and also docking, which is sort of loops us back to presentations from the last couple of weeks. So if you have used ChimeraX yourself, and especially if you spot some gaps or inaccuracies or things you think will be useful for folks to know either on the call or that might look at the video afterwards, then please do let me know during the course of the presentation or at the end. So I am going to kick off with essentially going through a list of visualizations and analysis to start with. Okay, I had planned to get PowerPoint alongside, but that's a bit irritating, isn't it? Let's get rid of that. Is that going to work for people, or do I need to make it full screen?
It's just about readable.
Just about readable. Okay, well, maybe I can, if it's just about readable, I'll stick with it. But if it's not, let me know. I have had nothing to do with ChimeraX. It's software developed in UCSF, and it's essentially available for academic use and for not-for-profit type things. There are licensing details on the ChimeraX download page. They talk about these two recent, this first very recent reference, and obviously encourage you to cite it if you use it. And as you can see, quite a few people have been involved in the development, and you may well know that it comes after Chimera, which they say is not being developed further. ChimeraX is the latest version and is actively being developed. There are probably a few things in Chimera that haven't made it into ChimeraX yet. There are definitely also some new features in ChimeraX and things like interface with AlphaFold. I'm just going to mute you, Maxime, if that's all right. Just a bit of background noise. Okay. If you want to unmute to ask a question, just let me know. Okay, so there's plenty of information about ChimeraX out there. As usual, searching online will provide lots of information. There is also some very useful help inside ChimeraX, which is, I guess, unlike some academic software. And in particular, I find the tutorials very useful. If you open up one of those from the help screen, you can click on the links. If you open it up from within the ChimeraX itself, you can click on the links and they are active. So you can essentially do the things live in your session that they demonstrate. I'm going to go through some of those things. There's also a nice presentation, recent presentation by Tom Goddard, the RSC setup, which goes through, I guess, perhaps a slightly more protein-centric view of the world than people purely interested in small molecules might normally look at, but it's very comprehensive in visualization methods. There's a nice short video on using AlphaFold from within ChimeraX to do your own structure prediction using the AlphaFold code. So that's possible within the current versions based on a sequence that you can give it. There's also this, if you prefer to follow along PDFs than videos, there's a nice PDF here which you can also take a look at. So plenty of help online. I guess I'm just going to go through some of the things about looking at a small molecule ligand protein that I find useful, and then we'll have a quick look at some AlphaFold things and some docking. So first of all, getting a PDB structure into ChimeraX. I should say that there's a range of the interface. There's a range of things that are useful. The command line down the bottom here, probably very small font. Apologies for that. That's why I've put some of the commands in the PowerPoint, which I'll also put a link to under the video and perhaps distribute to those of you that are on this call, if I can find you on our distribution list. And there's also the menus at the top, and also these icons under this series of tabs, which are useful. And you'll see in a minute that actually some of the elements from within the log are active as well. So if we start by typing a command to open protein, this is the crystal structure of Gleevec in its target protein ABL1 kinase. Then you can see that there's some information provided about it once we've opened it in the log here. And so you can do things like selecting the various chains directly from the log, which is nice, and also the ligand. It selects the 4 copies that are here in the crystal structure. You can also get more information. That's live as well. Maybe there's a bit of a delay because I'm doing a video session here, but usually that comes up with more information quite quickly. Doesn't seem to be playing ball now, never mind. So as I'm going to, as usually you'd be wanting to focus on just one copy of the protein ligand interactions. They're usually almost identical. I'm going to get rid of 3 of them, and various ways, as is common in ChimeraX, various ways to do that. Okay, you can see now finally the additional information's come up, the reference and things like that. So a quick way to get rid of the other 3 copies is use the command delete, and then the / and B to D. And what that's doing is it's getting the / means that what follows is a chain reference. So there are 4 chains A to D, and I'm going to delete B to D. And you can see here that there's other prefixes for models and residues and atoms and things. So that should get rid of all but the A copy. And just to put that more in the center, you can zoom in on my Windows machine with a mouse, or a mouse wheel that is, or just use this view command. So there is the ligand inside ABL1. I guess before we go too much further, it's useful to remember that this is an experiment, and we can look at the B factors, the temperature factors, and we can look at the electron density if we want to. So actually the B factors you can visualize on this very easily. Molecule display, and then you can color by B factor. So the blue are the low temperature factors, red are high, so the red is going to be potentially disordered. And one thing we can do is you can zoom into parts of the image with the graphics tab here. This is quite convenient. Side view, so we're looking at it front on. This is the side view down here in the bottom right, and this is a clipping plane, so we can clip away a good chunk of the structure if we want to just focus on a slice through it. And I'm going to try and focus on this red loop region and some of the darker blue B factor regions of the protein, because I want to just show that the link between that and the electron density. So we can open the electron density for this from an electron density server. So open to HYY, so the same PDB code from EDS, and that will bring in the electron density map. And you can see at the bottom here, bring it up, that we can move this slider to change the electron density cutoff that creates this grid view around the structure. And you can see that, not surprisingly, the temperature factor correlates with somewhat with the electron density. So the better resolved areas of the electron density, where there's more electron density, the temperature factors tend to be a bit lower. You can see this loop region, we haven't got much electron density for it at all yet at this threshold. So, okay, that's a bit of a diversion perhaps, but I think important to remember the link with experiment. I'm going to pull these clipping planes back out again and get rid of that. So what I'm going to do now, have a little look around the structure in other ways. One thing that's quite handy is there's a preset under here. If you get yourself in a bit of a mess with starting to do visualizations that are sort of going wrong, and preset and selections of various regions that are in a bit of a mess, this preset original look can restore things to somewhat near normality, which can be useful. And there's also an undo button, though it doesn't undo everything. Things like deletes aren't undoable. What else have we got? Okay, so I find it useful to separate out the protein and the ligand in this model panel here, so that we've got a copy of the protein separate from the ligand. And I find the easiest way to do that is with combine command. So on the command line, combine, and then use #1, which means combine model 1. So essentially we're going to create a copy of model 1, and you can rename these things so you can make things clearer as to where you are. So rename #1, and we'll rename that to the protein, and we'll rename #2 to the ligand, which is imatinib. And then we need to make that happen. So at the moment we've got 2 copies of the same thing. So I'm just checking this box here. So we're only looking at ABL1 now, the one that's labeled ABL1. We've still got the ligand in it. And now I'm going to zoom in a little bit and select and delete the ligand. So control click on an atom, and then you can use the up arrow, which is very helpful, and that sequentially starts to select larger portions of the structure. So one more click and you select the whole thing, and then you can down click and you just get back to having the ligand selected. And once you've got it selected, you can act on it. You can act on it to change the way it's displayed. And in this case, we just want to delete the atoms. So actions, atom bonds, delete, and that gets rid of the ligand. So now we've just got the protein, and we can now hide that and highlight the other copy. And now we're going to delete the protein and keep the ligand. One more thing that's sometimes useful, you can see that the center of rotation isn't close to the ligand, which is a bit irritating. So I'm going to select an atom in the ligand, again, control click and select it, and then under presets, I think. Maybe it didn't. Actions, that's where it is. Set pivot. So that's setting the center of rotation about that atom. Okay, so what I want to do is select this. Actually, I want to delete the protein, but one convenient way to do that, you can select what you want to keep and then invert the selection with select, invert, and then delete the atoms from all of that. Okay, so now we've got the ligand and the protein in 2 different models, and that means we can do various things with them. We can recolor them, for example. So you can recolor the atoms of the ligand. This is a little frustrating if you do it this way round, because you can see that the heteroatoms all become the same color as the carbons, which isn't what you really want. You can select the ligand, and so we could have, under actions, we could have colored all of the atoms. Having colored all of the atoms, if we now color by heteroatom, that gets us back the colors of the heteroatoms that you'd expect. In fact, this handy little command, original look, the way that works is, because we've now got two models, it does sort of vaguely sensible things and actually colors the carbon atoms different from the heteroatoms. So it achieves the same sort of thing, but quicker. And that you won't be surprised to know there's a command line to achieve all of that as well. So let's have a look. So now if we want to look a bit closer into the ligand-protein interaction, you might well want to see what's in the vicinity of the ligand, what residues are contacting the ligand. One way to do that is select the ligand again, and now select some additional residues that are close to the ligand, and you can do that with select zone, and now choose your distance. 5 angstroms is usually the default unless you've changed it. And you can also decide whether you want to select just the atoms that are contacting or within 5 angstroms of the ligand, or the residues. And I usually prefer to look at the residues, so I'm going to select all the residues where any atom is within 5 angstroms of the ligand. Apply just applies the command without closing the tab, or you can just click OK and it will do both. And now, so we've selected the residues that are close to the ligand, but we can't see their atoms. Under the molecule display, there's a show. So there's various things here for surfaces, cartoons, atoms. You can also achieve the same thing under actions, but this is quite convenient. So we can show the atoms of everything that's selected. And now you can see, we're seeing a bit more of the what's in the binding site. Just type the command view cell there, which puts the selection sort of center stage. And again, we can use the wheel to move things in a bit. We can also look at hydrogen bonds. I'm going to use Control and click in empty space to deselect everything. Again, you can click on one of these buttons up here, and they're also available through the menu. So this click in H bonds will generate a lot of hydrogen bonds, because obviously in a protein there's loads of them. You can see them all through the alpha helix, etc., etc. You probably don't want to see those. You only want to see those between the ligand and the protein for the most part. So again, if we select the ligand and then hit, actually I'm going to use the tools menu option for this. So structure analysis, hydrogen bonds, and now you get a menu option which gives you a bit more control over what you're doing with this command. So you get control over distances and things like that. For my purposes, I want to limit the selection where there's a hydrogen bond, where there's at least one end that's got a selected atom in it, and I want to select the residues involved in the hydrogen bonds afterwards, and reveal the atoms of the hydrogen bonding residues. So a bit more control over the hydrogen bonding command, and then you can see that this is now just showing us hydrogen bonds that the ligand is making with the rest of the structure. Slightly frustratingly, you don't always get to see the atoms in this view, because you can see here that this piperazine is making is making a hydrogen bond with a backbone of this residue, and the backbone atoms aren't shown. We can change that, I think, by hiding, no, wait a minute, yeah, so, yes. So we're hiding the cartoon display of all the residues that have been selected. And that kind of gets you most of what you want, but you'll notice that there's a disconnect here. I haven't got a better solution. There may well be one. Anyway, that does, I think, get you most of the way to what you want. One other thing I wanted to show is these surfaces. So you can create surfaces very quickly. This is solvent accessible surface by default. So you can see the protein, the ligand inside the protein there. You can make these, you can color the surfaces by various things, hydrophobicity, for example. We can also make the surface transparent, if I can remember how to do it. Surface transparency. Okay, so you can still see the surface, but you can also see through it somewhat. Okay, there's also, you can see the sequence alongside alongside the protein, which can be very helpful if you are wanting to select and manipulate particular residues and see, you know, what certain residues might be doing and how they're contacting the protein. So you may well be aware that Gleevec is kind of a first generation drug in this class. And we're going to take a look at crystal structure of 5-MO4, which has got nilotinib in it, which is a second generation compound. And there are ABL1 can create known mutations, or Gleevec can cause known mutations in ABL1 at 315 and 363. So you can go finding them in here, or you can also, what can be very helpful is there's a command where you can name a set of something, and I'm just, this is only 2, so it's not too bad, but you can imagine if it was more than that, it would get quite unwieldy. But what's very useful is you can name the residues that you want to group together, and this is position 315 and 363. 315 and 363. So that's given that set of residues, that pair of residues, a name, and then we can manipulate those. We can select them, for example. So user defined selections, now you can see that resist has come up, and that's the name I've given those. And you can see that we get the residues, and in this case it's surface. If I keep the surface on, I think for this purpose I might turn the surface off. On that. That's because I've got something selected. Right. So I need to select the, if you've got something selected, the actions are going to operate on the selected atoms or residues or sequences. If you've got nothing selected, then the actions act on everything. So when I did hide surface there, that did get rid of all the surface. So I'm going to redo that selection, and you can see that it's highlighted these, and we could, for example, recolor those so they stand out a bit more. Let's color them yellow. Okay, so those are the two resistance mutations that I was talking about. And if we want to analyze this further, we could then bring in a second protein, and that will give me an opportunity to show you alignment. So if we open 5-MO4, if we just zoom out a bit, that's obviously not aligned, quite separated. So we can use the command Where is it? Yeah, structure analysis, Matchmaker. So again, there are a couple of options here, but usually the defaults are pretty good. And we have to choose the reference structure. The reference structure is the one that's going to stay still, and then choose the structure that we're going to match it to, which is the one we just read in. So if we apply that, then you can see that we get the alignment. And if I turn off a few things, I'm going to select the Ligand again, really just so that we can get back to focusing on that. And then it's a little bit complicated. You can see the overlap between imatinib, the original Gleevec, and then the later compound nilotinib, which is picked out in green along with its protein, and you can also see picked out in yellow the resistance mutations. I haven't highlighted the structure for this one. If we select that, we can show the atoms of that residue in the nilotinib structure. You can see, so I have to show you the heteroatoms of this one to see that it's different. And color by heteroatom. Okay, so acid to amide. Okay, so that's what I wanted to do, just looking at starting to interrogate the structure of ligand protein structure. I was going to shift to a different session. So in Chimera, you can save the session. I'm just going to close it, but you could save it to a file that would allow you to bring back the entire thing. And I'll show you reading something like that in and how much more convenient that is. So this is a session that I've had before, which is related to this target, which is a malaria PDE. So there's this paper here from David Baker's group showing that phosphodiesterase beta is important in signaling in malaria parasites invasion. So there are no crystal structures of Plasmodium species PDE beta. So we're going to take a look at homology with some human PDEs and also at an alpha fold prediction of PDE beta. So this session, the CXS file, which I've saved previously, has got quite a lot of that information in. So we're focusing on the ligand, and a moment, if I first of all just let's deselect those. Okay, so this is a human crystal structure of a phosphodiesterase with a ligand, and we can, I'm telling you that there's an alpha fold structure. If we go to, you may know that alpha fold structures, a lot of them have been published and are available on the EBI website. Here, there's a lot of information about alpha fold if you haven't already dived into that, and we can search for, there's various ways to do this, of course. If we can search for phosphodiesterases and then narrow the search down to by species. Not every species has been published, but a lot of them have. And one of them that has is Plasmodium. So we've got Plasmodium falciparum. So now we've got just 7 phosphodiesterase relevant structures here, and we're looking for PDE beta, which is this one. So I'll have a quick look at the alpha fold prediction of this. And as you may know, AlphaFold has got a couple of different quality criteria associated with the structure. The first of which is this PLDDT, and that is a per residue confidence score. So it's really a sort of local environment confidence score. And so the blue is better than the red, and you can see that there's this region of the protein where AlphaFold is pretty confident in its prediction. And then there's these loop regions and just strands that it's got no idea about, basically. And helpfully it doesn't try to guess. There's also this somewhat separate region where there's quite a lot of blue there as well, sort of indicating it's reasonably confident about that too. But you'll notice that in this figure below, this predicted aligned error, this is where these you get an idea about whether the relationship between these two reasonably well defined regions is confidently predicted, and the answer to that is no. So this is the dark blue region where it's pretty confident that this sequence is well predicted and that the residues relative to one another are well predicted. But this separate region, which is the light blue one, although there are elements of reasonably confident prediction within that, the relationship between the two is not well predicted at all. So that's what AlphaFold thinks of the structure. And one way to bring that into to ChimeraX is to use the open command again. And in this case, you can see that it caches commands that you've used before. And I am going to bring in, here we are. So I'm going to use the UniProt ID. So we've got open, I think it's on the next slide. Yeah, so I'm using this command here, open Q816Z7, which is the UniProt ID of PFPDE beta from AlphaFold. And that has brought the structure in. And again, I'll just show you that structure on its own. And if I zoom out with view, then you can see that essentially we've just, not surprisingly, we brought in exactly what we've just seen on the website. And in terms of how that relates to human PDE, actually looks like it's already done the alignment, but we could use Matchmaker if that wasn't true. Okay, so I've previously taken a look at this. So what I wanted to focus on now is just having a look at the active site. So I'm going to focus on the ligand again, select that, and okay, I like to look at it that way up, and then we'll bring in the rest of the protein. So we've got two proteins here, so well, three. So this is the native crystal structure, which is a human crystal structure, homology of homology 2 PDE beta, falciparum PDE beta, and we can bring in the AlphaFold model and overlap it. So this is the AlphaFold model. There's quite a lot of, as you saw, it's a bigger structure with quite a lot of loop region that's not well defined. So I'm going to use the use this to get rid of some of the extraneous stuff we don't need to see. So that's the AlphaFold prediction in light blue of the PDE beta structure. You can see, for example, there's a sequence change here. So if you hover over, we've got asparagine to histidine. And I've also generated a Fire2 prediction of the same structure from this human PDE. We can bring that in too. So if If we just look at the Fire2 structure, then you can see the same sequence change, but now Fire2 has put this histidine in a different place relative to AlphaFold. One quite convenient way to view this in Chimera is actually to use command Morph, which shows you a kind of movie of one protein morphing conformation to another. So if we do Morph 2 to 3, which are the two PDE beta models, one from AlphaFold and one from Fire2, then Chimera should give us a movie of that. I'll leave the ligand in from the human PDE structure. So I'll zoom out a bit so you can see the movie. It takes a little while to compute, but then you should see the AlphaFold structure morphing into the Fire2 structure, which can be quite a nice way to visualize the differences between them. So you can replay that video, or you can just use this slider to get an impression of what's going on between the two. And quite helpful sometimes to just look at both. It gets a bit complicated. Let's see. Well, I think you'll get the idea. So you can see the regions of the protein where the biggest differences are between the Fire2 model and the AlphaFold model. And I guess one of the things about this, and why I perhaps picked out this histidine residue at the back, is that AlphaFold is making structure prediction in the absence of ligand. So it's not surprising that the AlphaFold model is placing the histidine pointing directly into the cavity where in the human PDE structure this ligand is binding, whereas Fire2 is taking the homology and saying, well, I'd be better off keeping this out of the way. So that's quite I guess an interesting difference between the two models. And if you follow this YouTube video, which is on the EBI website, one of the people behind AlphaFold talks about thinking about the AlphaFold structures in a similar way to APO structures without ligand, which I think makes a lot of sense. Just to maybe show you one more feature of Chimera. If you start with the human PDE structure, turn these off. You can actually ask Chimera to replace a given amino acid residue with another one. So I've inserted this bit of video in here to show you the swap AA command. I'm using swap AA interactive, and then you highlight the model number and the residue number that you want to change and what you want to change it to. So in this case, I'm going to change this residue here, and the interactive element gives you options here from the Dumbrack Rotamer library for Chi1 and Chi2 angles, and gives you the prevalence, so you can click through some of the more prevalent ones and see which one might be more relevant to you. And let's say we choose that one. You can also assign the right mouse click to various options up here, one of which is bond rotation. And if you right mouse click, then having done that on this bond, you can rotate the torsion, and similarly. So you can do further refinement if you want to, or just do the whole thing that way. Okay, the last thing I wanted to do, which loops back to the presentations earlier in the month from Professor Coase and Professor Durrant at the University of Pittsburgh on docking, is show you a way to analyze some doc output in Chimera. So first of all, I'm going to close this session again and open a different one. So we're going to open this one, which has got another PDE. This one's got sildenafil in it. I've already separated out the protein and the ligand. And I guess one of the things that you often want to do early on in docking is see whether in this particular protein, the docking software you're using does a reasonable job of redocking the ligand that you know does bind, because this is obviously the crystal structure. And I'm going to bring in PDB-QT output from Vina, where we've taken a look at that. So just read that in. Okay, so obviously that's a bit of a mess, just looking at all of those poses. This is 9 poses generated by Vina, all off sildenafil being docked back into this protein. And Chimera has a way to tidy that up. Tools, binding analysis, view doc X. And if we, so it opens a new window down here. If you click on the top line of that, then you're just shown the top by energy of the poses that Vina has created for you. And now you can use the arrows, the up and down arrows on your keyboard, to cycle through these different poses. And you can see that the top one of these, the lowest energy one, is actually not a very good fit to the crystal structure. The next one down is much closer, and that's not unusual in docking. So in this case, the second most energetically favorable pose, as predicted by Vina, is pretty close to the experimentally derived one. So I guess not uncommon. Then if we want to look at a larger set of docs, I'll show you that you can do that with Chimera. So a similar idea, we've got a lot of poses now of different molecules. And again, if we just bring those all in, we're going to see a mess. Again, we want to use ViewDoc to look at that. But now, if we just use the menu command, then ViewDoc X will combine the sildenafil multiple pose file and the different ligands where we've just kept the top pose. Actually, I think top 2, and combine them together. So instead of doing that, I'm going to use ViewDoc X on the command line and specify model 3, which is where this group of ligands is, for the ViewDoc X. And now we get another ViewDoc X window opening up. I'll close the one to do with sildenafil and hide it there. And now if we click on the top one of these, then we are getting one of the top poses from this multiple ligand file. So this is more like what you would expect from docking a large number of compounds into crystal structure. And again, you can cycle through them with the arrow keys and just take a look at the different compounds in there. This table picks up information from the PDB-QT file, and you can choose which columns to display. You can also sort by various things. Score is most obvious, I guess. And you can also plot some of these things, and you can write out a file. You can rate, make selections about which compounds you like. So quite a nice way to view some doc output. So in answer to a question about building molecules, there are ways to do that in ChimeraX. So if you want to do that, probably the simplest thing is to add hydrogens. I've put add H and then the model number #2, just so that we add hydrogens just to the small molecule of interest. And then you want to highlight the atom you want to change, the hydrogen atom you want to change. If you're building things, and use tools, structure editing, build structure, and then rather than starting a structure, which is how you could start a completely new one, it's a lot easier just to modify something. So I'm going to modify this side chain here, and we're going to change the hydrogen for a carbon, and you can see generally how you go. And then if we change that one for a carbon, I think it'll make a bond between the two. You can see here, connect pre-existing atoms if appropriate. Okay, so a little bit slow, but if you do want to make small changes, this looks fine, and there's delete options and bond building options as well.